So if you make it too general, such as maybe genetic studies on Drosophila, okay, so what are you doing with Drosophila, right? So, but what about maybe genetic studies like or of body color pigmentation in Drosophila or wing development? Okay, it's probably more specific, but you could make it even more specific, right? If you talk about some of the results, for example, you could mention something um, like mode of inheritance of your field. Maybe it's controlled by a receptive autosomal gene, right? Or body color pigmentation is controlled by a recessive autosomal gene or a dominant sex point of view. So just tell me what your paper is about. So it doesn't really have to be long, right? I mean, the more informative you want it to be, I mean, you have to include more, you have to make it longer, but just try to make it concise, right? Give information about the phenotype it's controlling, and maybe the mode of inheritance, like the dominant, etc. So once you do that, then you have the abstract. And the abstract generally you would write this actually at the very end when you're done with most of the section, then you would write the abstract. It helps you kind of distill down all all the information that you have presented. So then you can summarize. So generally, abstracts are written at the end, and put in the beginning of the paper, but generally so you can write them at the beginning too. So an abstract then is, instead of, we said for the title, it helps the reader decide whether or not they want to read this paper, and this is like in real life. And this, and in real life, you want to show that make you so this might be interesting. The next thing you want to know, they're not going to read the entire paper, they can read. But abstract is like a summary of what happened, what's fine. And that will help them decide, okay, is this the paper I'm looking for? Should I spend the next 30 minutes reading it or not? So in abstract, if I asked you, like to give you like a summary, like give you done in a few minutes, it's well, we have to understand or we can apply this specific thing or mutant to see how what effect, to see how that dominant is excessive, is extensive. So and to do that, what we did is what we have cross-wise that we and we did the reciprocal cross to see if there are differences in the sexes, right? So, and our results have showed that our mutant the human is recessive and is on from X and is that from X, Y, and Z. This is like, so that is, so you want to make sure that you explicitly mention what the objectives are. Make this bigger. So the objectives are 1, 2, and 3. And to achieve those, we did X, Y, and Z. We did crosses, we did PCR, we did extraction. I mean, you will not talk about DNA extraction in this paper, right? You talk about what we crossed, the mapping process, how they were done. And then the results, which is right here, we found X, Y, and Z. You should be direct at first time. So, and then you can have a concluding statement that shows that this is kind of summarizing the results or what they mean. So say that this is an autosomal recessive gene. You don't necessarily have to add any type of future studies in the abstract. You could, but generally they go into the discussion. So if I have, I would start with my abstract with like a bare bone abstract, having some information about the objective. And you could probably find that you only have 20 words. You have still a little more left. Then you can add more information. You can expand the method and then expand the result. Expanding not necessarily just to make them big, but to allow more information, right? Instead of just, if I only had 100 words or 200 words, I'd say we did crosses and reciprocal crosses, and then we did mapping crosses. And then if I had 400 words, specify exactly what crosses we have made. And if I have more room, I could actually continue, right? Depending on how many words. So if you still can, just expand and add more information, because the more information you have in the abstract, it's going to be more informative, as long as it does not exceed 350 words. So, but you want to make sure that your abstract, like for example, that you don't have room to explain. If you mention a few methods, make sure that you address part of the result for those methods. We did one, then we did two, and then we did mapping. And then you come to the results, you talk only about the process. Wow. So why did you mention it in the first place? Okay. So make sure that you mention the results, both the methods and the results section. So if you tell me what methods you have done, well, you should tell me what the results are for that thing. So as you expand, just make sure that you are having some type of balance. If you increase the methods, also make sure to mention the results for those experiments that you have done. Any questions about the abstract? Okay. I think generally during this class, we're supposed to go over next week's material, but I think I'm gonna make sure that we spend enough time writing and also in lab because unfortunately, this class is one of the very few classes that you guys will, in which you will study writing. That's something not ideal in our department. We encouraged that there would be more writing like courses at the beginning of your college program. I know Dr. Medina's class, a little bit of writing. It was only this class and also Dr. Pat. So I think that's why I want to make sure that you get some feedback, you have a chance to ask questions, and get enough training in writing. It's just one course, but we're trying to do our best. So if you have any questions about abstract writing, anything of writing assignment 3, make sure to post those on Canvas or ask them in class. So for the introduction, the reader has kind of read about exactly what we're trying to do, what you have done and your results. So then the introduction then is kind of a gradual development of your project. So in the introduction, it's giving you a little bit background of what I'm trying to do, why should I care? So in the introduction, we'll talk a couple of sentences about Drosophila as a model organism, and you will talk about your phenotype of interest, body color, pigmentation, wing size, bristles, and you will talk about the wild type gene, like what does it do when it's not mutated. So for example, I talk about my vestigial wing, like wings that are developed at this age, and there are a bunch of genes developed with F1, ebony, singed, or black. So you would talk about the phenotype, the body color, the wing development, and you don't really need to go to town on this. A few sentences about the phenotype, and you would talk about what it actually does. So if your gene is a MET kinase, if it's what it actually does in gene. So like what you guys have done in writing assignment 1, where you propose 5 genes that if mutated would lead to your phenotype. Right? You talk about those 5 genes. So you would talk about your gene, you know that my gene is turns maybe this turns X into Y and the Y. When it's mutated, if this gene is mutated, so this reaction will not happen, leading to X not being converted into Y. And that would lead to the ebony, vestigial, singed, or black. Right? So here you want to talk about the process in which the wild type gene is involved. What does the phenotype, the wild type phenotype look like in your trait? Wild type. So we'll talk about your specific gene. So then you would say when this gene is mutated, it leads to that phenotype. So which is this one, right? So here we'll talk about the normal wild type, what happens, how color or how colors develop, or how the wings develop or bristles. And then we'll talk about when there's a mutation in your gene, what happens. Well, what happens is actually the opposite of this, not getting the phenotype, I'm getting something else. Because like you talk about the molecular, what happens. Right? So, and to make this more interesting, when someone's talking about flies, maybe not everyone would be interested. So we try to make talk about something with ortholog. So what's an ortholog? What's an ortholog? It's a gene that is very similar to your, have the same sequence, very similar sequence, and has the same function, right? In another species. So if I you want to open Flybase, how do I find like an ortholog of my gene? So let me type vestigial in here. So and you would find that there's actually a lot of useful information, general information, genomic location, functions, summaries, variants, phenotypes, orthologs, and I think there should be also references down here somewhere. So this is orthologs. I click orthologs, and it shows you that those are the genes, the human ortholog. So this is all of these VGLL2, L. So those are similar genes in humans, right? That have the same sequence and probably the same function. I just picked this one. So 9 out of 14. Those numbers are the number of different databases that predict that this gene is similar to the human gene. So those are different, I believe, different tools, online tools that would find ortholog. So I would probably go with the first one. And so the human Homo sapien VGLL2 is your human ortholog for vestigial, for example. So I could type, maybe click on this and see what comes up. It's called vestigial-like family member, right, in humans. So you could go and find if there are any diseases, human diseases, associated with this gene. Disease, so it says spindle cell, rhabdomyosarcoma, some type of cancer. So I would look into my gene and find a paper, not just Google or any type of AI. I find a scientific paper that talks about this gene, like this one, for example. That's not a paper, I think that's just a summary of the gene. It tells you what the gene is. It's a protein with a transcriptional enhancer factor. So what you want to find is just find a couple of sentences about this. Is when you find it in your paper, you want to make your gene more interesting, but in the software it's going to go in. It is human too, right? Or especially the human ortholog. So just a couple of sentences in your introduction, just to link it to the big picture and have some relevance to the human genome. Any questions about orthologs? So if you can't find any orthologs in humans, you can take orthologs in other species. So this is humans, orthologs, humans over here, other organisms. If you don't see anything in humans, you should be able to find something. But those are the different genes in rats and mice. So okay. Now that you have told me how important your work is and what happens when it's mutated, and how is that equivalent in humans? Okay, now I see this is important. Okay, what are the objects? Let's say, well, the objectives of this research were to want to distinguish some examples located on, and specifically what on the chromosome. Right? So here you want to be very explicit. You don't want to beat around the bush. This specifically tell me exactly the object. So that's what the reader is trying to know. You don't want them to think and try to guess what your objectives are, right? You tell them 1, 2, 3. Okay, I know that you told me why this is important. You told me what you're going to do. But I'm expect to tell me how you did it. On the materials and methods, you would talk about what you have done, the crosses, all of that. And I think it's generally useful before starting directly with cross X and Y, or these mapping problems. Before jumping into the details, it's generally helpful to give the sentences roadmap. Like maybe something like this project had maybe 3 phases. The first phase was the crossing, which we did like the mapping process online, and then we did actual lab work. Right? So you want to talk about the 3 different steps. So when you tell me what you're going to do, the method is going to be structured. So tell me as a reader what to expect. Instead of just going directly to crossing fly, collecting versions, and then we did online process, and then we extract the DNA, and then PCR, and we'll get lost. Reader, what is it? Give me the big picture. What are the big steps? So crosses. So in this paper, we don't really have the extraction or PCR. It's going to be mainly the mapping process, so the actual W21, W22 fly process, and then mapping process, online mapping process. So this is what you told the Then you would move to the next section where we will talk about the actual process, what you did, what kind of process you have done. So it's generally advisable when you talk about anything that you've done in lab, you cross the wild type with the mutant to find out. You will start with the one, but in order to find whether the phenotype is bound or recessive, okay, you cross it. In the same thing, we use this fancy machine, right? We use PCR. Well, Prometheus, what are you trying to do? Okay, in order to learn X, we did Y. So in order to figure out whether the phenotype was white type female or male, and to determine if it was sex linked with the reciprocal one, you would talk about larval score. You say you waited 10 days until the fetus, and you don't have to use passive language. Maybe 400 flies were counted or were scored, and the F1 was observed, right? So when it comes to, when we talk about those crosses, make sure to mention only information that are enough for the reader the same. So it's something important with the incubation, something such as if you use antibiotics, maybe the content of the antibiotic, and generally in the freezer they should be used. That antibiotic should have the concentration, how much the concentration is. So we should information that are necessary for that field, because we will need in the colors of the food or the like a lot of information that's not relevant and not necessary. So all the information that is enough for somebody with your knowledge to do therapeutic experiment. So when it comes, so you can mention that the food, maybe we use the food for lineup biological like the company. When it comes to the mapping process, again, you would say in order to determine the recombination frequency and the distances between unknown genes and other genes, we cross this one, A, B, and C. So make sure to mention the Y first. So what else are we missing here? Any other questions? Any? Yes. So the question is for WTM1, WTM1, WTM2. Do you need the fly numbers from FlyLab.js or from the actual crosses? So this is from the actual crosses, right? So WTM1, WTM2, we use the actual numbers that you have counted, right? So for mapping crosses, you have to use FlyLab.js data, right? But for WTM1, WTM2, after the question. So you could also mention that if we have the statistical analysis we did using R or whatever we had, which is mostly R. So we also talk about in mapping across A, B, and C, we scored maybe 1,000 flies or whatever fly is, whatever number it gave you, right? So some students find it easier to talk about these processes as if they have done actual process. You can do that as well, and as long as we know that you didn't do it yet. But if you find it more easier and it flows easier to get your actual process, go for it and explain that it's the same. Or I think I got a question before about should we use the name of our mutant, like the nickname, or should we use the actual gene name, Ebony, flag, Mysterio, or Cindy? And again, it doesn't really matter, whatever flows. Sometimes it feels a little strange if you talk about that the gene is ebony, vestigial, cinch, or black, and we want to determine its location. Well, we know the location, right? So sometimes it makes sense to use the nickname. Sometimes it could make sense to use the actual name. But if you pick one name, just stick with it through the entire paper, right? For me, it doesn't really matter whether you use the nickname or whether you use the actual name. So what have you guys been doing? The nickname or just the real name? Real name? Or you have not started writing yet? Okay, yes. Stick with one thing. Okay, so the results now is where you would share. There's like two things: DBM1, DBM2, and WTC. This was something that we did not do. So we had students do double balance of processes and the full process, but we didn't talk about this. We didn't do this. We didn't have to worry about it. We would just get the points. So these are some 3 points. So in the results section, we talk about the results, right? Which is you show the results for WTM1, WTM2, and the genetic basis and rationale for this. The way I would explain this, well, since I've already talked about what I did, what type of process work we have done, we would start here again by reminding the reader of the objective of what we were trying to do. But I would briefly touch on the objectives. I would not repeat and explain exactly what is it that we have done because we've written the message. So you could say WT1, which was which we did, which was done to figure out the mode of inheritance, showed F1 was phenotypic dominant. Or so the rational and genetic phases is mainly the phenotype that we show that it will be the dominant phenotype. Now, on how two individuals we're expecting that the dominant phenotype is the one that we show in the F1, right? This is like the rationale. So you would say maybe 400 fly, 400 of them were wild type, or all of them were wild, or some of them were wild type, some of them were mutant, case of thing. So we would show the numbers, and you don't really need to that this one is dominant and it's heterozygous, and our mutant is recessive. You don't need to explain any of that here to discuss it. Just tell me what the numbers that you got in the F1. So because of the results, we generally only can offer any explanation. We don't offer any type of interpretation. The same mapping process, the rationale would be that genes that are farther apart from one another would have the frequency, right? And closer, they would have a smaller percentage of the rationale. So the outcome that we had those numbers, Metaplus, we had those numbers, Metaplus, we had those numbers. So, and of course, we'll have tables for those method process at the end of the table. So whenever you put results in here, make sure that you show the results and not to the figure. The information is present in both the tables. And it's being redundant. So this is how we show it. You don't want to put information and then say as seen in figure 1. So the differences between the Whitehead and the Newton are as seen in figure 1. And not tell me what those differences are. As you have to explain what those differences are. Yes.
For each of the tables, we also need to have a description explaining that.
Yeah, so the tables, do we need a caption or a description or a legend? Well, you need obviously a title, and titles and tables go above the figure before the title, before the table. So you don't necessarily need like a caption or a legend for a table as long as there's a descriptive title. So you don't really need to have a caption or a legend or more explanation for a table, as long as your title is enough. If you really need to, you can. So yeah, but generally, if you have enough, if the title is enough, then there is no need. Okay, we go here for the results. For the mapping crosses, you would mention like how many the results that you have gotten, and the mapping crosses that you have, what those results are, and then you can also repair to the kinetic map, right, that you have designed or designed to. I put a figure of the kinetic map. So up to this point, we haven't really discussed any of your results yet, right? In the discussion section, you would explain, you would offer some interpretation. Something as simple as that the F1, the scored F1 was heterozygous, like the F1 individuals were heterozygous, or the fact that you crossed two embedded lines, two pure lines. So the Whitehead and the Newton were both pure, then were homozygous. And it was dominant, which shows you that this was Whitehead, which shows you that. And that's about it for 71 and W2. In case if you had vestigial or not, because it's a sex-linked trait, you'll probably explain a little bit more as to why you start different different categories. And you'd explain like X-linked chromosomes, and you'll explain the phenotypes you got in the F1. Okay? So then we'll talk about any sources of error. If you had any problems in your analysis, maybe if you didn't have enough lives to score, if you only scored 100 or 2 or whatever, then that's the reason it's over. The F1 was not as expected. And if you had 400 wild type and you got 400 wild type, but you also got 5 mutants. Instead of killing those and just not reporting them, say that we found 5 mutations, like 5 mutant phenotype, 5 mutant 5, which is unexpected. Probably that's sad or what. You didn't put your members' names, right? You can just say that we had a problem or we think that this is what happened. So you offer some type of explanations for any issues you have in mind, or the results are not as expected. In the future research, again, this could be a paragraph, a couple of sentences about what the future work would be. So now that our paper doesn't talk about any of the molecular work, gene extraction, PCR, or mathematic analysis, you could say, well, the future work would include extracting DNA from both wild and mutant, and doing PCR, both doing sequencing runs to exact or to determine what the mutation is. You have references, and we're using the genetics journal references, which is similar to APA. So where you have the authors, the names, author names, then the year, and the title of the publication, and then the journal, like page, volume, and issue. So figures you would have would use the same convention and that you did in writing assignment 1. So you could use the same figure, but make sure that this figure is exact on your writing assignment 1, and you'll see on the figure. Make sure to improve the figure. So it will have a title, a caption, put some arrows, stuff that you want to highlight. So figures again should stand alone. So I don't really need to go back to the text on its own with the title, legend, explanation should stand alone. I should be able to understand what you're trying to say. tables, you would have the tables that you are including, and you will have tables for writing assignment one, WTM1, WTM2. So you have these two tables for the numbers that you have scored. Then you will have the other tables for mapping across A, B, and C, the actual number. So I think this was, you can include, it's not really required. I think this was from the past semester, so we don't really need to include the calculations, because in real life, you don't really include chi-square calculations. We did this in writing assignment 2 as practice, but we probably will not include it. You could include, if you want to, please do, but generally it will probably not add them to the writing assignment. You could instead use any type of p-values. If there's some calculations, you could include the p-values in the text. And there is, of course, 10 points for general writing, which is the formatting of the paper and the quality. So let me quickly look at the questions that you guys have submitted. Hopefully that this explanation, oh, why is this still oh, my screen is sharing. I'll disconnect this because I'll look at your submission and it will have your names, so I don't want to show your names with the questions you posted. Yes, yeah, maximum similarity score. I would try to keep it under 15%, but it doesn't mean that if you are over 15, you are in trouble, or if you're less than 15, you are in the clear, right? It really, as long as somebody did not copy from somebody else's work, like sentences, you should be good, because the percent would differ based on the length of the paper, right? So if your paper is one, if somebody has a paper that is 1 page, right, and they copied just one paragraph from someone, well, the similarity percent would be like 40%, 50%, because that paragraph is half the paper, right? But if somebody copies that same paragraph and their paper is like 30 pages, well, the similarity score would be maybe 3 or 4%, like very low, because that paragraph out of 30 pages is nothing, right? But when they copy one full paragraph or one full sentence, so those numbers, they don't really mean anything, but for us, it kind of helps us to look at where the high percentages are. So as long as you did not copy somebody else's work or most of the paper, but I'm doing little changes, I think you should be fine. So if you also copy from your own writing assignment 1, 2, or 3, it could increase it, may or may not increase it, but that should not be a problem. So you could have 20% similarity and still be okay, right? Somebody could have even 3% and could be problematic. So I would look at the similarity reports. Once you submit your assignment, you should be able to see the similarity reports, yes. Yeah, so for the figures, they go at the end of the paper. After the references, you would add the figures and tables. And make sure to cite them in text, yes.
Can we not embed tables and figures within?
So figures and tables should not be embedded within the text. They should be separate at the very end.
Might be changes, and there's a cycle that I go to do. That constant dopamine.
Mhm.
Like, oh, since this dopamine is now finishing this, it would like
Is that in the introduction? So generally, unless you're writing a review paper or something that explains, unless you're talking about something that is not new, something that is known, you should be able to explain and express that. Figures would be really awesome if you want to show how pathways are happening. If this is something that you have discovered, right, or something that you are providing a new insight, but it's generally not advisable. An image is worth a thousand words, right? It's better to have an image. But generally, if this is not your image, right, you're taking this from another paper. So generally, we advise against having those within the text. And I don't want to be to deceive. Who's your TA? What section? Oh, Aziz. Okay. So yeah, when you publish, so when you have the problem is with having pictures within the text, it makes the formatting a little weird. Some pages have not much text, and in real life, in journals, they would have a lot of the figures within the text, because journals have specific formatting. They have 2 columns specific. They have specific format that makes those figures look nice and look okay, right? So, but in the reports or writing assignments, I think we've agreed to have them, all of them, at the end of the paper. So, and I'll make sure that all TAs, we are all on the same page. So, but I'll make sure that we're all on the same page. You guys are being graded the same way. It's not like my TA wants this. Because our TAs, they come from different backgrounds. They have, some of them have PhDs. So Aziz has a PhD, Sita. So, but at the same time, we want to make sure that all of you guys are treated and graded the same way and held to the same expectations. So we will make sure to do that. We grade papers. All the TAs, as I mentioned, they would grade one specific paper. All of us, including myself, would grade it. And then we come to the board and plot the grades. Aziz, what did you give it? Emilio, what did you give it? And we'll see what the variability within TAs, where that variability is coming from. So make sure that there is no such variability as my TA wants this, and make sure that we're all on the same page. Yeah, short answer figures go in at the end of the paper. Okay, so let me look at, so this is questions about, are there any pressing questions? Any question that I did not answer? Okay, and I'll include, yes, go ahead. Is there a question? The what? Sorry, go ahead. Page requirement? Yeah, there's no really page requirement. You could submit a 15 pages or 12, 13 pages and should be fine. You should submit it. You could submit a 20 something pages, could be fine. We're not deducting points for having short, like smaller paper, but you could lose points for missing information, right? And the same goes for writing essay. If you have a longer one, okay, it's fine, as long as you're not adding extra fluff that is not doing anything, right? Some stuff that tells me that you're not aware of the audience. Right? If you start to explain, maybe this applies more to writing assignment 4. If you explain how things are work, you don't really need to know that. But there's no penalty for, like, we don't really have a specific requirement for the length. As some of you said, feel free to leave if you need to. If you have a class, I'll have a recording too, but I'll continue talking. So some of you had asked it that things feel repetitive because in the abstract, you will have the objectives, methods, and results. Then again, the same will happen in the results section and the method section, and it's okay for things to be repetitive. So yeah, you don't really need to include calculations, and also you don't need the R code. You'll need to include that in discussion as well. You have discussion figures. So yeah, whatever is on the rubric. So in the discussion, you can talk about the meaning of the results, like what it means, how we came up with those mapping process, the mapping distances. I think in the past we had a discussion about, you could actually, let me see, I don't want to add something that is not on the rubric. So yeah, we're not really, in writing assignment 4, we'll probably discuss that more, but here is not really, you mainly discuss the results. WTM1, what does it mean? Mapping process, what does that mean? Thank you.
Professor, can we use APA instead of?
I think you can. You can use it. It's very similar to the genetics, and you can. Yes. Took attendance for writing assignment. Okay, let me stop the recording first. Writing assignment 2.
Yeah.
Present, and you were counted absent.
Okay, good morning, everyone. I think we'll go ahead and get started. I hope everyone is having a great afternoon. Hope also that you have some relief with the writing assignment being extended for 1 more week. I have to say, like, this semester is all different. All the TAs I have are new, except one. I think Nathan is a returning TA. And when all TAs are new in one semester, that kind of puts more effort on me. And also sometimes labs are not delivered the way that I have wanted to. So, but we're trying to make it better. So to make sure that you guys get enough instruction on writing, we'll go over writing assignment 3 today as much as we can. And in lab this upcoming week, you will review one of your peers' writing assignments, like writing assignment 3, and give them feedback. And similarly, you would receive feedback on yours from one of your peers. And whenever you're doing so, do this using the rubric. Right, look at the rubric and evaluate your own paper or your peers' paper, and of your TAs being in lab, ask them questions. I would hope that TAs are always proactive, and many of them are. They would ask and reach out, but they don't always know exactly where you are struggling, especially if TAs did not take this class before. So if you're struggling somewhere, by all means, let the TA know. Right? They can't really know where you're struggling. So please let them know. So come up with a specific question. You can ask the kid, how can I make this writing assignment, like this abstract? How can I make this better? What is missing here, right? How would you improve this? And instead of just asking general questions, how can I improve my writing assignment 3? I just have specific questions. So the plan for today, we'll go over writing assignment 3 rubric, and we'll probably look at one of the examples. So I'll answer as much as, I'll go through it to make sure that I covered all that stuff. And then maybe in a few, maybe you'll have maybe 20, 30 minutes to the lecture. I will open a quiz, a place for you to submit questions about writing that I have not answered yet. Okay? So if you have any questions, just wait a minute until I go through the rubric and see if you have any questions. I can also make it open at the beginning. I'll probably go through the assignment first and then we can talk. So, and one thing I don't like about the biology course like structure in this department is that you don't really get to practice a whole lot of writing in the biology department until this stage. I know in intro Dr. Medrano do some writing, stuff like that. But there's not really a whole lot of writing in the biology department. So it's probably maybe genetics lab is one of them. I think there's also the biochem with Dr. Patterson, I believe. So writing assignment 3. This is the writing assignment 3. You think of it as first draft of writing assignment. So writing assignment 4 is the final theme. So writing assignment, whatever you have in writing assignment 3, you will actually use it again in writing assignment 4. So it's not like writing assignment 4 is a whole new assignment. Right? The time you spent in here, that will pay off. I think I'll share with you writing assignment. The rubric for writing assignment 4. Writing assignment 4 should be here, nothing simple. So this is the rubric for writing assignment 4. Download. So in writing assignment 4, you will see. So writing assignment 4 is all of these contents, all of those stuff. Those are stuff that will come. So those are the exam. So the abstract, see the object. So this is literally copy and paste from writing. So those green cells is what's new in writing assignment 4. Right? So you're writing assignment 3, we'll talk about the flies, done. But we didn't talk about DNA, PCR, or the right? That is not included. So that will be included in writing assignment 4. PCR, blast, and the equipment that was used, right? So in the method section, you will add the DNA, PCR, and stuff like that. Again, in the results, you will use the same results from writing assignment WT1, WT2, the mapping process, but you will add the molecular section of it. The same goes for the discussion. So whatever time you will spend in writing assignment 3, that would pay off right in writing assignment 4. So you can literally copy section from writing assignment 3, use it in writing assignment 4. Of course, after considering feedback that your TAs will give you a writing assignment. So back to writing assignment. So the title, just a descriptive title that would tell me that is specific enough. For example, if I say studies on Drosophila or studies on mutant Drosophila, it's not very descriptive, right? But I could say, what about, I'll tell you a few options and you tell me which one is better, right? Studies on Drosophila flies, studies on mutant Drosophila. Body color is controlled by a recessive gene, by black, a recessive somatic or autosomal gene. Or you could have maybe something like, which one of these do you like more? Last, right? It's kind of specific, tells me about the gene that is controlling the phenotype. It tells me about the nature of the mutation, which, like if it's that it's a recessive mutation, you could actually include the location. You could say a chromosome 3, include the chromosome location. You wouldn't necessarily write the number of the chromosome. Like you would say it's at 3.71, right? You could say it's located on chromosome 3. You could, not necessary, but make sure that if there is only one sentence that I will read about your paper, that's one single sentence that I will, which is your title, should be as descriptive as possible. And you don't really have to make it very long, but just try to make it. I mean, it's difficult to have it be short and informative at the same time, right? They go against one another. But just try to be concise and have the least amount of words that would deliver the information, right? So the inheritance of the trait, what it controls, or even what it does, right? I could say maybe black body color, or maybe a recessive allele or recessive phenotype controls a MAP kinase, or that codes for a MAP leads to vestige results. or if you could include the mode of action of the gene, you could, you don't really have to. But the title should be descriptive, tells me what the mutation is, what it does. At least the bare minimum that it tells me what type of mutation, what body part are you talking about, right? Bristles, wings, and stuff like that. Okay, so if there's one sentence I will read that should be the title, should be descriptive. Okay, what if I have more time? If the reader, if your reader is able to read the abstract, if they have the time to read the abstract, well, that's good. I have more words to say, right? So let's combine and distill your work into an abstract, right? So in the abstract, if you ask me what the book is about, I'll tell you, okay, the objective of this research was to figure out whether the mutant phenotype was done successfully or not. And also to do that, I did some crosses. I crossed the wild type with a mutant and a mutant. The second cross to know some system. I did that, and then to figure out whether the chromosome is located, where the mapping crosses cross. The results have shown that F1, when I cross X and Y, I go for the stage or wild type, which shows that the mutant and the mapping crosses showed that ebony is located on the chromosomal tree and position X, Y, and Z that far apart. So it has a couple of pieces. It has the objectives, like what are we trying to do? What are we trying to do? What are we trying to accomplish? And then how did you about doing it? What did you do? What did you find? Right? So methods, results, maybe I could do instead. So you want to make sure that it's balanced. So because you don't want to talk too much about the methodology and then mention stuff that you will not talk about the results. Right? If the abstract, if I will say that mapping crosses X, Y, and Z, and did this cross ebony with 3 other genes, right? You spend too much time like words talking, but then in the results, you don't talk about the results of those experiments. No. If you mention something that methods, well, you have to talk about the results. So I would have a balanced abstract. The methods, the results. If you did that and you still have more words, you can add more. But make sure to win this field, right? If you add more to the methodology, you also expand the results and also expand the discussion of the conclusion. So if you add more to the methods, well, you have to talk about the results. The reader will feel, okay, something's missing. You told me about stuff that you did, but you didn't know what the results are. So this is the abstract. And you can generally write it at the end of the paper. Once you're done with your paper, you can write the abstract. And people generally do that. When you're done with your experiment, then you can write your abstract. And the most important part of writing is starting, right? Just have it, open the document, just dump anything, write everything, anything, right? So the writing process, your paper will not be like that. It's not gonna be perfect, never going to be perfect. No one's writing is perfect, right? Just dump anything. Don't worry about that commas, that semicolons, don't worry about it, don't worry about the grammar. On the first draft, just dump anything, right? Copy from other papers, not to copy it as it is, but maybe this is an idea I saw, or style of writing I saw it that I'm gonna not literally copy, but I will use. I like it how you introduce the topic. They start with a strong topic sentence, and then they started to explain. So I like that stuff. I could copy that and put it into my paper. Not the final draft, of course, but you don't even have to change the format. You can just use it just to remind you that this is something I wanted to do. I wanted to copy this stuff. Right? And you can copy something successfully that they successfully have done, but not literally copy the exact test. You can copy the test, put it on your first draft just to remind you of what you wanted to do. Right? So I saw maybe somebody elegantly transition from an idea to another idea. Right? So I like that. So I tried to copy it. So, but not literally put it on your final paper, right? So just getting to write anything. I wouldn't worry about commas, about semicolons, words. I wouldn't worry about that. I just have the ideas there, right? And make sure that once I have everything, random stuff, right? Then we'll organize it. But I make sure that I put everything in it. Then the next step would be to go over the paper and then start to have some structure, right? The introduction, then I have a bunch of stuff that could go into the introduction. But in my second, and there's not really a first, second, and third version, right? You're continuously editing and changing, right? It's not like specific discrete steps, separate steps. So in the Drosophila overview, in the introduction, is where we are telling the reader like why he or she should care, right? What we are doing. So we're starting with just a broad review of Drosophila. It's kind of cool, fly, like how it's useful. And then we can talk about, you don't really have to go to town on this, maybe a couple of sentences, what Drosophila has been useful. Discuss the biological relevance. We'll talk about your phenotype, right? Blood, like maybe the body color, wings, whatever mutant you're talking about, right? So we'll talk about how mutant Drosophila is, what you're studying, right? And how we could talk about maybe body color in Drosophila is controlled by several genes. You could give the reader some background about the wing development or bristles, how that is happening. So whatever comes to your mind is just write any idea, just to have something, right? And then you can modify this later. So, but don't think about having the perfect paragraph. Just write anything, right? And then so we're talking about the biological process. What phenotype are we talking about? Right? The wings, the bristles, the body color. So, and then you will talk about how this gene generally functions. So I'm saying maybe if my gene is body color, I will talk about how body is developed, how body color is developed. We have several genes. We have ebony, black, ebony. I have tan, I have brown, yellow, several genes in that pathway. And those work together. Even I don't really understand the exact mechanism of every single gene in the pathway. That is fine. You could mention that these genes, that this phenotype is a complex phenotype controlled by multiple genes. And one of the genes that we're interested in is SING, or if you're studying wing development, right? So one of the wing genes is vestigial, right? That's what we're studying. And you can use either the actual mutant name vestigial, ebony, singed, or black, or you could use the nickname, whatever feels natural and normal and fits. It's up to you. Some people find it easier to talk about the actual mutation happening. Some other folks feel that it's easier to use the nickname as it is, as it were, and whatever feels more comfortable and easy to. Okay, so I will talk about the gene and why it's interesting. And if there's a human ortholog, if there's a similar human gene, I will talk about it. I said ebony is the gene. Well, the human ortholog, what's an ortholog? Yes.
Something that we have that carries over something similar to that fly.
Yeah, so a gene that is similar in sequence, not only in sequence, but hopefully also in function. Because it's only similar in sequence that would we would call it homolog, something that's similar, not necessarily in function, right? But the ortholog would be similar in the sequence and also in the. How can I find a human ortholog? Two ways. You can take the protein sequence of your protein, go to BLAST, do a BLAST to find. So let's go to BLAST, not protein, do protein BLAST. We'll talk about this upcoming week when we talk about Arabidopsis, but let me give you a lazy way. Flybase. You can go to Flybase, find your gene, vestigial gene, robot. I will go over here, gene models, products, orthologs. So this information is really actually very helpful. So this is the summary of the gene models and products, tells you how many, this is the phenotype of your gene, right? What types of alleles we have. So, but down here you will see orthologs, shows you human orthologs. So all of those are genes in humans that are similar to you, right? So you could just have ebony, singed, or black, any one of those, right? So I think those would be, maybe you take this one because it has 9 out of 14. And 9 out of 14, I believe those are different 9 databases that predicted that this is an ortholog, like different websites, bioinformatic database that predicted that. So this is the human ortholog, BGLL2. So when it comes to BGLL2, you want to make your paper interesting, right? Maybe somebody was reading, who cares about the vestigial and flies? Maybe not many people, but if you say, well, this is actually the ortholog of BGLL2 in humans, which when mutated causes this disease, right? Makes it so this is vestigial, like you can search this gene and see what it is. You can see disease. There are specific disease, include spindle, trouble, something, coma, related to cancer, and pleiotropic, whatever. You will see what that is, right? You'll figure what disease is that, right? So you'll see that it's spindle, so it has something to do with the, you will see exactly what it has to do with plasmodium. You'll have to sarcoma, related to some type of cancer. But you'll see exactly what the gene does. Okay, so you say, my gene in humans does this, and when it's mutated, it causes this. Yes, sir.
Would you need to define like what an ortholog is, or would you assume the reader would already know?
That's a good question. Do you need to define what an ortholog is? No, because assume that the reader is a geneticist or somebody who's familiar has a decent amount of knowledge. So I wouldn't define PCR or so imagine that the reader is one of your classmates, so they should know what it is. That's a good point. So I put what the mutation is, how it affects the function. So I talked about my mutation, talked about the ortholog. Make sure when you talk about your mutation, you mention how that mutation has an effect. I said if my gene, if my mutation, my allele does X and Y, it turns X into Z, right? So we have the normal. When this gene is mutated, well, then my gene, which is the MAP kinase that turns X into Y, now X is not turned into Y. And now I will not have my phenotype. Instead, I'll have the vestigial black epidemial syndrome. One simple statement as this should do. So, okay, now that you have told me what you're studying, why you're studying, the human realm, now explicitly tell me exactly what you're going to do. Exactly, specifically, explicit. So something like, okay, now that you give me an idea of what you're trying to do, why, and why you're tell me what is it that you're going to do. And explicitly say, you should probably could state what the objectives of this research is to 1, 2, 3. Be very explicit, right? Don't just beat around the bush, just explicitly, right? Is to determine whether the mutant phenotype is dominant or recessive, if it is sex-linked or autosomal, if it is which chromosome and where it's located on. Don't let the reader guess, because if you leave some ambiguous, well, they will think. I don't want them to think what you are doing. Just tell them what you're doing, right? So the objective is to 1, 2, 3, 5. You can't miss this, right? So any paper, when you're reading any paper and they're having a bunch of stuff, you're not really knowing what they're doing. You're like, for God's sake, tell me exactly what is it that you're doing? What are you trying to accomplish, right? Yes, sir. What's your name?
Sebastian.
Yes.
So how do you make it seem like you're not repeating exactly the objectives you just put in the abstract? Because I don't know, that sounds like you're just rewording.
Yeah. How do you make it repeated in the introduction without making it sound repetitive? Well, it will be repetitive. You will exactly repeat them because these are like two different audiences. Because in the abstract, and when it comes to reading a paper, when you guys read papers, when you read the abstract, you are deciding, is this good for me? Is this a paper I want to invest some time reading or not? Right? So the abstract should have all the components. Right? In the introduction, you will repeat the objectives, but you could elaborate, right? You could increase. Then can I elaborate and have more details if you want? Not necessarily, but if you find that the objectives you have in the abstract are enough, fine. If you need to add, you can, but don't feel obligated, right? Just to make for the sake of making it big. So yeah, and throughout the paper, there will be, and that's okay. So, but don't feel like you have to come up with a new way of rewriting the objectives, so it doesn't sound repetitive. That's fine. Although you could do that. Yes.
Okay, so I just want to make sure, because how I wrote this, like, for the most part, you just have to hit these main broad points and that pretty much your points. Given that you're not going very little into writing a page on each point on the page.
Yeah, I don't want to say like the word points. I really want you to be able to write, to improve your writing and write something that makes sense, like beyond this class. Yeah, the point is not really having an A, B, C in this class. To be honest, wouldn't really matter once you graduate. And unless you're trying to show, I mean, nobody looks at your transcript. So that's, and when you apply for a company or somewhere, they're not going to say, okay, you got a C plus or whatever in this specific class. Nobody honestly cares, right? Unless you are applying for a genetics position and you have C's in all of your genetics classes, right? That probably would. But generally, having a B versus a B plus versus an A minus, and I like that you guys come and fight for points. That's totally right, and I used to do this as a student. But if I'm missing a point, I just go talk to a professor. But when you graduate and get your transcript, it's not really that significant, if at all significant. Okay, so the most important thing is to be able to write and convey and express yourself. And this is what I wanted to help you to do. For the grades, I think I mentioned this before. Half of my students will get at least half would get an A. I mentioned this before, right? I don't know if I mentioned this. I think this was one of the comments I got from the dean that you're giving students A. Who cares if they did what they're supposed to do and they learned? Why would I just lower them for the sake of having a bell curve? So okay. So just focus on writing. Okay. So explicitly specify the objectives. Specify the objectives, 1, 2, 3. What questions do you guys have about this before we move? So, common question I get, how long should the paper be or the abstract? As long as it needs to be. So, we're not really measuring, so length is not part of the rubric. And if you're submitting a paper, I mean, many journals, all of them, they have specific requirements, specific lengths, specific pages. As long as you're keeping your audience engaged, right? Doesn't really matter. As long as the person who's reading is engaged and keeps reading, if he's entertained, that's fine. If you can make them entertained without being bored, right? Because if you put too much information, if you start explaining what an ortholog is, and maybe take a paragraph to explain that, and I know what an ortholog, I feel bored, right? I feel like the writer is not aware of the audience. So, but I would say most of the papers I've seen are anywhere between 13, 14 to maybe 20 pages. And that is including several pages for tables, a couple of pages for paragraph, page for references. So it's not like you have to put 20 pages of text, right? So again, I hate to say a number, but because students keep asking, I just want to give a rough estimate of most of the papers that I have seen. I've seen papers that have 10 pages and they're awesome. I have seen papers that are 20 pages and they're awesome. So like length is not really fair. As long as you have points for longer paper, unless you become not aware of the audience and start explaining stuff that are not needed. So now that your reader is ready, you've told them that what you want to do, what the objectives are. Let me now take you on to how I'm going to do this, right? What the material is. And before jumping into the material, we crossed this and we did this and this. Just take a minute and give you really like a roadmap, right? This will make more sense. So I would say, well, this project had 2 phases. One was actually 3 phases. One was the actual crossing on the flies. Then there was a mapping process component, which was done online. And then there was last portion, which was done maybe like in the lab, molecular section of the course. So you could just give the reader some type of, because the readers are generally lost. The only one who perfectly understands the paper is you. Right? I think most of you peers and TAs and me, well, we will understand what you're trying, because we know what you're trying to do. But generally, a reader doesn't really know what you're trying to accomplish. So just take your reader like a baby, like tell them, okay, here is X, here is Y, here's what we're trying to do. So having that overview, right? You don't really go to town, just tell them that this project has 3 stages. We did X, Y, and Z. Just give them a simple role, a few sentences, 3 or 4 sentences. So when I see scoring flies or crossing fly, what is that? Right? But when you tell me that you first cross flies, then you did online, then you did PCR, I'm expecting to see this. So when I see crossing flies, well, he told me about that. She told me, so I know that this was coming. So just 3 to 4 sentences, just to prepare, because you don't want any bumps in that ride, right? You want this to go smoothly. So whenever you do specific material, when you introduce any type of material, anything that you have done, You would say we crossed the wild type and the vestigial. No, don't tell me what you did and then you explain me why. Tell me why. Tell me the why first, right? I would say in order to determine whether the mutant was dominant or recessive, we crossed X and Y. Right? I wouldn't really care what you have done unless you tell me why. Why should I care to listen? Right? So in do X. So instead of just telling me we did PCR, gene extraction, we use all these cool equipment, now tell me first, in order to isolate the wild and the mutant alleles, we did PCR. So tell me why you did. So start with in order to do X or Y. And then tell me what you have done. So for example, for WT1, in order to figure out whether the mutant phenotype was dominant or recessive, we did WT1 in which we crossed the vestigial with wild, right? And to figure out whether the mutant was sextant or not, we did WT2 in which we did XYs. And then we scored flies by looking at the under microscope. We counted 200 flies. Gabriel?
I was going to ask quickly the stack materials about this. Do we have to mention that we used the fly lab for the process for the BMT process?
That use the fly lab? Not necessarily. I would assume that you use some type of lab.
The online thing.
Yeah, the online, yeah, you should use that we use FlyLab.js.
Yeah.
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 Let me open the quizzes. I see some. Let me open and make questions whenever I assignment 3. I'll continue, but I see some of you are holding the backpacks and they need to leave. So let's see. Questions. What questions do you need? Yes, let's serve this as 37 questions and questions will be the same. This is the same. So let me make this open until this open from today. So put this under this week's modules, which is the week of the 14th. And you can go and submit any questions over October 14, which is this quiz, right? Assignment 3, right? Assignment B, should be available under the week of October 14. Can you guys see it? Anyone can see it? Okay. So if you have questions posted about the assignment B, I'm going through the assignment in order. But if there are any urgent pressing questions, I can also jump to these. And I'll make this recording available. So posted there, and this will count as attendance. So if you have any questions, just type in. Okay. I'll speak for maybe 5 more minutes and then I'll start taking questions. So that's your question. Okay, so let's continue then. So once we're done with the materials, we cross X and Y. So you don't really want to put in the results. It's just the material section. In the results section, one error here is the described outcome of DBM1, DBM2. That is not, shouldn't be there. This is something we canceled from previous semesters, just used to do another process that we canceled. So in the results for writing assignment 1, WTM1, I wouldn't say WTM results were I got 100% wild type, or well, they probably have forgotten what you're trying to do. So you can repeat, and again, that will feel the kind of Sebastian. You'd say, well, in order for WTM1 was done to determine whether phenotype was dominant or recessive, and it showed that we got 100% wild type. And so just slightly remind the reader what you're trying to do without making it a long statement, because you're not trying to show them how to do it, right? Because you already did that in the methods. But now you're just reminded of what you did, not necessarily how, but just remind them. So to test whether the results of WTM1, which was to figure out which is whether the is dominant or recessive, the results have shown X, right? So I would try to be just remind the reader, but make it short. Right? So WTM1 had 100%, while T2 had 100%, right? Or whatever results you got. It's generally 100% wild type. I wouldn't explain this shows that this is dominant, this shows that this is autosomal, and this is why, and here is why. No, you save that for the discussion. So in the results, you just tell me what numbers you got, right? What you found, not necessarily what it means and why. Right? So we don't really discuss. So if you said, okay, I got maybe 200, and which shows it's dominant, okay, that's fine. But generally, I would save any explanations to the discussion section. So again, the same goes for mapping crosses. you say that the distance between unknown and A, it was X, unknown and B was that, unknown and C was that. Without necessarily discussing the mapping distances and the genetic map and how it looks. So in the results, generally we talk about the results, the numbers, not necessarily what they mean. What they mean, that goes into the discussion. So the results section is generally short, not very long. So in the results, make sure that you reference, if you show a table, if you said that one or the mapping cross is mapping cross A, we got maybe those numbers. Don't tell me numbers are as shown in figure 1 or table 2 without mentioning what those results are. Mention what the results are and then refer me to the table. Send the reader to the table or to the figure. And so if you talk about the phenotype, the differences between the wild and the mutant, you could say that you should not only, if you said that the differences between the wild and mutant are shown in figure 1, that's not enough. Because you're just sending the reader to figure 1 without telling me what's in there, right? So you should tell me what is it, what the result is, right? What is it that you're trying to compare in the figure, table, whatever you have, just tell me what it is. Tell me in the text. And when I go to that figure or table, I should also be able to understand that, right? So when I go to a table or figure, I don't really need, I shouldn't have to need to go back to the text. The figure and table should stand alone. So the figure should have enough explanation. If I needed more info, I could go back to the text. But the figure should have enough information to stand alone, to give me some basic information. Okay? So in the results, if you refer to a table, a figure, or something, just mention what the result is. Tell me what the table is trying to tell the figure. And when you tell me that message, I will go to that table of figure and I have your message in mind, right? I know that it shows this, and I'll go to the table. So don't leave lastly in the discussion section, right? You will discuss, you would explain more philosophical, more deep analysis of what's happening, right? If I have cinch, for example, say I got different kinds of males and females, wild type and mutant. And this is because it's a sex-linked trait, and this is some genes or sometimes you have maybe X, you have big A little a or big A little a. Males would have this phenotype versus females they would have this. I have more of this happening. That explain, show me some genetics, right? It doesn't have to be long, but just offer some explanation. If you can do it concisely, good for you. So source of that's where I would explain any issues, right? If you got something unexpected, right? So if you got, maybe you're expecting white, maybe it will F1 if you have the stitch black, but you got a few flies, something that you didn't expect, right? Instead of killing those flies and acting as if you didn't see them, right? You should still report those data, right? And offer an explanation, say probably, maybe Megan did this, or maybe Mike maybe was killed. You don't really have to mention the names, but say maybe there was a contamination. I don't call anyone on the paper. I just found professionally generally. This could have went wrong. This could have happened. If you don't have any sources of error, don't feel like I have to make something up to get the points, right? If you don't have any sources of error, we'll give you the full points for it. Don't feel like you have to search for yourself. References, figures, tables, general writing. Those should be easy. General writing is like the quality, organization. So let me move to the questions that you have submitted because names will show. So I want to mute this. I have like a million buttons and you can't really find the one that you want. So on screen, okay. Okay, so let me look at the customer regression. I'm not really sure when this will show up again if we need to. You don't necessarily have to mention that there was no errors. You don't really. But if you didn't mention any errors, I'll assume that you didn't have any errors. Why does this show up? Why does this show up? Okay, so question analysis. Okay, some of the questions that you guys have, open this. Okay, question NA, okay, no question NA. Should we include chromosome map like one in the WA2? That would be great. Yes, please do it. So when we read, we can see what's happening. And go into the discussion. No questions. For the results, we're going to add. So for writing in the results, you don't really copy writing assignment 2 with its format and dump it and writing assignment, right? Just pick the table that you needed. Maybe write mapping across A, B, and C, right? Just the numbers. So you don't really need the code, the ARM code, the chi-square calculation. You don't really need any of those, just the numbers, right? So the question, we're using Flybase. How do we cite the information? So you don't cite Flybase, but you look at the paper that you have. So for example, if in the ortholog section, you see a paper, a specific gene, right? I would find a paper that talked about your gene. On Flybase, there is a section that talks about this actually. There should be references, but I will not cite Flybase. I will find the actual paper. There's a paper that says VGL2 or your ortholog is related to this disease, right? I would go and find that paper. I wouldn't cite Flybase. Okay, no question. And for the results, yeah, we don't copy anything from copy running assignment to us 3 or 4 table, 5, whatever tables we need, not just copying the entire records. So we're using Flybase, we will cite Flybase. Do we need a picture of the chromosomal map? Yes. How do you incorporate the assignment project in assignment 2? How specific how specific should the abstract be? Should fit as much information as you can within the 350 words, as long as it's balanced. And what can we ask if we don't understand the biological process of a gene? You can ask Google. So you can Google to understand the biological process of your gene. Google can talk about, if I'm looking at body color pigmentation, I would type body color look at the genes, see what is happening. Adenine, white, studio. You can talk, you can email me, we can talk about it. You can talk to me as well. So, other question, because you shouldn't be explaining too much of the vocabulary, because you would expect that your reader knows those terminologies, such as orthologs, PCR. So, you shouldn't explain things that are obvious that the reader knows. In the discussion section, should interpret the results. And you can also discuss future research in the discussion. At the end of discussion, future research will do PCR and DNA, the stuff that you guys like. Assignment 3 put it as future research, right? Because it's not listed, it's not reported in the paper yet. It doesn't have any DNA or PCR analysis. Okay, folks. If you will need to leave, please do. I'll make this recording available. I'll continue. So, and how should we send the sources? The genetics field, you shouldn't cite the lab, no, book, and you just said that we document this stuff. In the direction, you talk about the source of the soft block, antibiotics. You can talk about the concentration of antibiotics, but not necessarily the sources of antibiotics for the companies. If you need to list this, the fly food source, you could. That's a company called Carolina Biological. Sounds good. So you could list the company's name, Carolina. DBM1, there's no DBM1 or DBM2. Method section, how detailed should the method section? Should be detailed enough for somebody with your knowledge to be able to replicate it. So if you don't tell me what you've done, then it's not sufficient. Should definitely put the PCR primer, not PCR. Well, not doing PCR now, but tell me the incubation time, incubation temperature, how many days. So it should be enough for somebody with your knowledge to be able to replicate this. Think of your group mates, right? What information? So if something is missing, something major, you didn't tell me the temperature of incubation, right? That's something important. So that's how detailed. Figures, again, you should probably have the same figure you had in writing assignment 1. Of course, after considering any feedback, you put better titles, make it a standalone with description. Will there be any extra credit for writing assignment 3? I think we'll think about it. Could be, maybe. It's not a big problem. What's WTC? Yeah, WTC, we didn't do WTC, so I will not work. Yes, listen to the rubric, but it means that you will get extra points. Something we didn't do and everyone will get it, so that's like DBM1, DBM2 that we canceled, you guys will get credit for this. So the specific format, APA, MLA, yes, we're using, so MLA is mostly used in humanities, we're using the APA. It's mainly not specific, we're using actually the genetics journal citation. It's kind of similar. So if you Google genetics journal, it's like this the same way, like you have the year, the author, so the author's year, title of the publication, year, volume, issue, and page in the journal. So, okay, how many tables? I would look at the tables are worth, each table is worth 1 point. I think you have a table about the actual crosses you have and the mapping crosses. So, if you, I would look at the maybe like 5 or 6 table, I believe. So, no any comments. Thank you. Difference between results and discussion. I think we mentioned this. So, which one should be longer? Not necessarily longer. Generally, discussions are longer. It's not like, okay, let's measure this. Your results is shorter than the discussion. You're in trouble. No. So just the results, just report the results, and the discussion explain. And if you've explained what the results mean and it is short, fine. Right? If you have done so and it turns out to be long, fine. Right? There's no like this must be longer than this. So what do we do about it? Well, you email me. So if they don't respond, sometimes they don't respond, but I'll try to respond. So yeah, if your email doesn't respond to me, please reach out to me specifically. I see you can directly reach out to me. Since we are not doing the DBO process, then it will just get 3 points. Should the figures go after references? Yes, figures and tables go after the references. Should we in-text citation, of course. If you have a specific piece of information that says this gene is a MEP kinase that does this, of course, tell me who said that, right? Mike et al. Or Michael Johansson, whoever, right? That's short form. And then at the end of the paper, you will put the full reference itself. Yes.
The chi-squared tables for us to, I guess, like in the ends part, right? Like,
Yeah. So the question is, do we need to discuss the chi-squared values and tables and details? Not necessarily. You could just say the p-value was X, which shows that this gene is it's linked or it's within a mapping distance, but not necessarily like you have to put a bunch of numbers or tables, calculation. Just tell us what the p-value is and what it means. Okay, how do you effectively transition without making your writing choppy? Well, in the paragraph, whenever you're transitioning to the next one, well, your paragraph should prepare the reader for the next one. So at the end of the first paragraph, you should prepare them. So for example, if I'm writing about, I don't have a specific example in mind, but to make things less choppy, the new information should not appear abruptly. You should prepare for it. So in the previous paragraph, just tell me something about what is coming. So for example, if I said that we determined that my gene was my phenotype was sex-linked, so the next step was to determine. So then we further sought to identify the chromosomal location. That's the end of the paragraph. So the next paragraph, I'm expecting that we're talking about the chromosomal location. And so in the first paragraph, you talked about the dominance and recessiveness, and whether it's sex-linked or not. And then you said, and therefore, or the next step was to figure out or to determine the chromosomal location. Okay, I know what is coming. So in the next paragraph, in order to determine the genetic location, we did this X, Y, and Z, and we found that X, Y, and Z, right? So if I wanted to move to talk about human, like the human ortholog, right? If I wanted to switch, and I talked about this gene, in my next paragraph, I talk about human orthologs. At the end of the first paragraph, this gene, what it does in Drosophila house, and I say, and this pathway is relevant to humans or has some human relevance or relevance to humans. Okay. Well, the next paragraph, I said the human ortholog of this gene is X, Y, and Z. Right. So at the end of the paragraph, I would prepare the reader, right, that you will switch gears. That you will make it less choppy. One way to make it less choppy. No question. I was skipping questions that are repetitive. Okay, that's the end of the questions. What other many questions I did not answer? Yes, sir.